Now, this slide lists a number of factors that contributed to the crystallization of beta-2 adrenergic receptor GS complex. Now, the first factor is the discovery of a high affinity agonist named as BI167107. Now, the binding affinity is the strength of the binding interaction between a ligand and a receptor. And binding affinity of ligand is influenced by non-covalent intermolecular interactions such as hydrogen bonding, electrostatic interactions, hydrophobic and van der Waals forces between the two molecules involving the ligand and the receptor. The characteristic feature of the high affinity agonist was that it had a dissociation half-life of more than 30 hours. Now, this long dissociation half-life ensured that most of the agonist would stay in the bound form with the receptor during the crystallization process. Because as I previously mentioned, that the presence of different structural conformations in ligand-bound GPCR was a hindrance to obtain a pure crystal. So use of this particular agonist with a very high dissociation half-life that ensured that most of the ligand would stay bound to the receptor, stabilizing a particular structural conformation, ensured that these researchers would get a very high quality pure crystal that would provide a very high resolution diffraction pattern, and that will be very helpful for determination of the structure. The second factor was the removal of GDP during crystallization process by using apyrase, which is an enzyme that breaks down in GDP. The third factor was the use of a detergent, which is known as MNG, which was designated as MNG. The use of this particular detergent ensured long-term storage of the G proteins and the GPCR, as well as it aided transition into a liquid cubic phase or mesophase during the crystallization process. Now, the detergents, their main function was to stabilize beta-2 adrenergic receptor and GS complex during incorporation into the mesophasic lipid, which was used for crystallography. Sorry. Now, in physics, a mesophase is a state of matter that is intermediate between liquid and solid. And biological structures such as lipid bilayers of cell membranes are examples of mesophase. And mesophase is very important for stabilization of membrane proteins, as well as, and that's why it is very useful during crystallization process. So this group of researchers also used a new mesophasic lipid called 7.7 MAG to accommodate the G protein during the crystallization process. Now, the 7.7 MAG structure is provided here, which has a long hydrocarbon tail attached to glycerol moiety. Now, in addition to all these factors, the use of nanobody and amino terminal T4 lysozyme also facilitated the crystallization process. So I previously explained the scientific basis of the use of nanobody and T4 lysozyme in great details. So this slide is showing the active state of beta-2 adrenergic receptor structure. The left panel is showing a complex involving beta-2 adrenergic receptor and NB8T nanobody complex. And the right-hand picture is showing a complex involving beta-2 adrenergic receptor GS, G beta and gamma proteins, as well as nanobody 30, which has stabilized this complex in its active configuration. And as I mentioned previously, that T4L was a fusion N-terminal T4 lysozyme fusion protein, which actually facilitated the crystallization process of this active beta-2 adrenergic receptor GS complex. So again, here I have summarized the four major factors that contributed to the determination of the crystallographic structures involving beta-2 adrenergic receptor and trimeric G proteins. These factors are ultra-high affinity agonist, which is BI 167107, detergent MNG3, nanobodies NB80 and NB30, and T4 lysozyme fusion proteins. So finally, by adopting these multiple approaches or strategies, the inactive and the active state of beta-2 adrenergic receptor was revealed. Now this figure is showing a comparison between the inactive beta-2 adrenergic receptor and active ligand-bound beta-2 adrenergic receptor. The inactive state structure is shown in gray color, and the active ligand-bound state is shown in green color. And both these structures were superimposed to show the relative shift in the conformation following agonist binding. As you can see that the agonist binding to the active site caused a 14 angstrom outward movement of transmembrane 6 and a 2 angstrom inward movement of transmembrane 5. Now in the next few slides, I will explain how this outward movement of transmembrane 6 caused a conformational change in the trimeric G proteins. Now this slide is showing the crystal structure of inactive and active beta-2 adrenergic receptor complexed with trimeric G proteins. In the left panel, you can see that beta-2 adrenergic receptor bound to it trimeric G alpha beta gamma proteins in an inactive state. And in the G alpha subunit, you can see that there are two domains. One is a RAS-like GTPase domain, and the other is a alpha helical domain. And the GDP binding site is occupied at the interface of these two domains. Now the middle panel is showing the active beta-2 adrenergic receptor following agonist binding and still complexed with trimeric G proteins. Here again, all the proteins are color coded so you can see their relative position. And on the right panel, this represents the superimposed crystal structure of the active and inactive state, and that clearly indicates the conformational change of the G proteins. Now in the middle panel, following coupling to the high affinity agonist, the G alpha subunit undergoes large structural changes, with the alpha helix being displaced approximately 130 degrees, and the carboxyl alpha 5 helix of the RAS-like domain is displaced 5 angstrom into the core of the active receptor, and this stabilizes the active state of the receptor. So this displacement of alpha helix as well as more subtle changes transmitted from the receptor to the GDP binding pocket through the beta 1 strand of the RAS domain, and these collectively responsible for the dissociation of GDP. Now the empty state beta-2 adrenergic receptor GS complex is then poised for activation by the GTP. So once GTP binds to the G alpha protein, then the whole trimeric complex dissociates, and this subsequently goes and bind to adenylyl cyclase or other downstream effector proteins and initiate the signaling cascade. Now this slide is one of my most favorite model explaining the functionality of beta-2 adrenergic receptor and agonist binding. This figure provides a concept that agonist alone does not fully stabilize active state. Now I will explain it in more details. The left panel is showing beta-2 adrenergic receptor and is representing only 2 transmembrane domain. Now this figure is representing different structural conformations of the beta-2 adrenergic receptor under basal activity, and there is a dynamics that is present here. So that means that no particular conformation was favored, and the whole structure is depicting a spectrum of conformations which are constantly shifting. But when agonists chemically interact with the receptor, then a particular conformation or a set of conformations were favored over the others. So here you can see in this figure, the number of conformations has decreased compared to the conformations present in the under the basal condition, basal activity condition. And then on the right panel, you can see that when there is a regulatory protein, for example, a G stimulatory protein is present, then together the regulatory protein and the agonist favored one particular conformation over all other ensemble of conformations. So what it means that agonist alone is not capable to stabilize beta-2 adrenergic receptor active configuration. Both agonist and the regulatory protein or a stabilizing protein is required to stabilize the active configuration. Now, previously I have mentioned that following agonist binding to the beta-2 adrenergic receptor, the G-alpha subunit undergoes large structural changes with the alpha helical domain displaced by approximately 130 degrees. The mobility of the alpha helical domain was confirmed by cryo-electron microscopy. Now, the cryo-electron microscopy is a technique which involves imaging frozen hydrated specimens at cryogenic temperatures by electron microscopy. Now, the specimens remain in the native state without the need for dyes or fixatives, and that allows the study of fine structures of protein complexes at molecular resolution. Now, here in this series of pictures, you can see the beta-2 adrenergic receptor complex to trimeric G-alpha proteins and nanobody 37 as well as a T4 lysozyme attached to the N-terminal. And by comparing different structures at different angles or positions, it was confirmed that there was a 130-degree shift of the G-alpha helix in the active configuration of beta-2 adrenergic receptor. Now here in this video, I am showing you a molecular simulation of the dynamics of agonist binding to the active site of muscarinic receptor. This animation was created by Professor Arthur Christopoulos' lab at Monash University, Australia, and this is a 30 microsecond simulation. You will notice a movement of the transmembrane domain 6 outward movement following agonist binding, which I have previously shown you in case of beta-2 adrenergic receptor. Let's play this video. Now, one more time, I'm showing you to highlight the dynamics of different conformations or structural ensembles in this molecular dynamics.